Yes, ma'am.
Just have to do it twice. Okay, let me get back to something that I glossed over in the genetic algorithm for alignment. And the background really is the independence assumption. The independence assumption being that what I do at any position is independent of what is at the other position. Where does this assumption actually come in for alignment methods? It comes in various places. One is on the whiteboard. One is not on the whiteboard. Yeah, well, actually, two of them are on the whiteboard. The hidden Markov model is on the whiteboard too, yes. And the hidden Markov model is a little bit more complicated. The hidden Markov model is the algorithm that depends on this assumption. Otherwise, you cannot guarantee that you find the optimal solution. Do you see another place on the slide? Dynamic programming is the same thing. You cannot guarantee, if you violate that assumption, you cannot guarantee that the dynamic programming finds the best solution. That is what this matrix here, the dynamic programming matrix, is indicating. Now, for BLAST, you need the background statistics. The best background statistic, again, you simulate using single letters, and you simulate using that assumption. The moment you violate that assumption, the complete estimate of the background distribution is wrong. The e-values become wrong. Okay? And you all know this is not true. And again, so here's an example where we see that in the alignment there's a sort of a correlated switch between positions, or at least some dependency. Highlight the charges here. We clearly know. Now, the great thing about the genetic algorithm is it does not need that, because it sort of matches segments. It operates on segments. Here's an example of what you can do with a genetic algorithm. There is, from the NASA, is a spacecraft antenna that is actually this thing here, and the shape of that one here is optimized by a genetic algorithm. And you see that the shape that you see looks pretty close to random, right? It looks pretty close, like something that you will not immediately recognize as, that looks nice and round, that may be the solution. It's just the opposite of that. And in fact, that is surprisingly non-trivial to find a solution. The way it does it, you essentially start, you have an initialization of the population, you compute the fitness. How would you do that in the alignment? That's essentially the objective function, right? The objective function could be, for instance, in dynamic programming, the simplest one is, what's the simplest objective function you can have to measure the similarity between the two proteins? Yes. No, you're sort of tricking me a little bit. I ask you what is the similarity measure, and you say distance. Yes, distance is 1 minus similarity or something. But yeah, no, but what exactly? What exactly do you measure?
Is it a sequence or?
Yes, we were looking at sequence alignments, yeah. Yeah, that's the simplest one, right? You measure the number of identical letters. And that then is a fitness. Fitness is, you put in any pair of sequences together and you ask how well they fit, okay? When the fitness is above some criteria, you say stop and you end. If not, then you have certain operators. So you can do, we begin by rouletting parents, meaning choosing parents at random. That's what that means. Then there's a crossover to children. So you cross over parts of that parent and part of that parent and inherit, so to say in this particular case, we have 3 amino acids. Now, how would you do that for alignments? How would you create the parents? Yes, but listen, what we're trying to do is, again, I'm sorry, I did not set this up very straight. I'm repeating a story that I was too fast at, I felt, and I'm again fast. So I'm not really good at it. But the major problem in the background here remains this one. We know all of the methods we use, hidden Markov model, BLAST, dynamic programming, everything that everybody in this field uses to compare proteins, sequences, has the independence assumption. And we know it's not true. So what system can we come up with that sort of gets over that? And genetic algorithm is the solution, but now what we want to do is we still want to align two proteins. So we're not looking at different homologs. We have two proteins that we want to align. Just we want to mingle with the alignment. We want to align them slightly differently. Or multiple proteins, but let's begin with two, so to just illustrate the problem. So where do we get the parents from in this alignment problem? Yes, but that so again, initialize the population could be a randomly chosen population, but is that really what you would do in this field? So it would work, but the point is, I did give you an idea, or I did mention at least last week, you may remember, that this is compute intensive. So the further away from the solution you sample, somehow the more likely it takes longer. So something that is already long, this is a surefire way to make something that is already long a little bit longer. In principle, it should work.
So you start with sequences that you already know that are very...
Again, I want to align two proteins. Ultimately, the strength of tea coffee of the genetic algorithm really is for multiple alignments. But just to get to the heart of my question, all you need to imagine is two proteins. Where do you get parents? So the parents are in fact not the proteins. What are the parents in this game? You want to change the alignment.
Two possible alignments.
Exactly. That is exactly where this could be the global alignment and this could be the local alignment. This could be with one gap penalty, this could be with another gap penalty. And you immediately, remember there was always this problem how to choose gap penalty, how to choose between global and local. There were so many choices. And now suddenly you have a way to forget about these choices, just throw them all in, right? And then again, you choose parts of the alignment from local, parts of the global, or you simply match and change it. Then you do the mutation. You compute the fitness again, and then you iterate over that. And those are again the people who did this. Now you begin with a bunch of alignment methods, and the cool thing, and I'm still. So I worked out this example in a little more detail, but I'm again not going through it. But the point is, you in fact the secret behind it is you consider everything and it can find the right solution. And that in fact here is the right solution. The great thing is that you can, on top of having parents that are from different types of alignments, you can also throw constraints in the objective function. So in the fitness function you can say, well, the fitness increases if I actually match a cysteine bond, which again, cysteine bridge any contact, because I and J are independent for alignment methods, you cannot align constraints that come from binding. And here you can do that. Okay, that binding constraints is in fact one for structure, but it's also for binding sites. So it's very important for function too. Tea coffee. SS search is somehow a similar idea. It's a sequence profile comparison, made faster. Ultimately, we are still here with sequence, pairwise sequences, sequence profile. We somehow can intrude into the twilight zone. And the question now is, what can we do beyond that? So there's sequence comparison, pairwise sequences, the sequence profile comparisons, where you have sequence compared against the family. What's the step up to go further? Finding more distant relation. What could you imagine? Come off it. Just in terms of names. And what is the next one? Yeah. The funny thing about science, very often in the simple things there is a solution. Profile alignments, exactly. Now, profile alignments in principle are are very clear. So rather than just having a sequence compared into this, you now compare two profiles. Very, very clear. And then you do that on a larger level, so you do one profile against many, and you have a finding. Now, the complication comes simply from blowing up the parameter space. And now we get into essentially what you just said, is the Euclidean distance. You can compile a vector product in different ways. You have two columns that you ultimately compare. And you can do that in many different ways. You can compile a Euclidean vector distance, you can compile an added sum of differences, you can compile a correlation, and these differences unfortunately matter a lot and blow up our parameter space. It also is something that may not always be wanted. So here is an alignment again, where you see that the way I construct that, and the colors are in this particular case not charge oriented, but you see that you have three that behave in some way, another three that behave another way, another two that behave yet another way. So averaging over that position is what you would do through the profile, is actually maybe not what you want. Maybe what you would want is to maintain this, let's call it structure, right? That, where I call structure is the wrong word here. I mean, really don't mean protein structure, but the grammar is maybe the better word here, where I say that if you are most similar to those three, fit into that. Most similar to that, fit into that one. And by going profile, you actually have a tendency to generalize much beyond what we see in a local environment. So it's not necessarily always wanted. Now, the other thing here, what we want to start with, is a sheep hack. A sheep hack to profile, that is a hack that allows me to do something without actually addressing the complexity problem. How could you imagine that I could do a sort of pseudo profile alignment? So I do not want to do sequence profile. I cannot do profile. I want to avoid at this point profile, because then immediately I have to decide how to compile Euclidean distance or what. And I don't want to have that, face that question.
You do one-on-one sequence alignment from each, a sequence from one profile with a sequence from the other profile, and so on.
Yeah, but then every comparison is a simple sequence profile comparison, right? But that's exactly what you do in a multiple alignment of sequence profile. That's exactly what you do, right? That is what I presented in cluster W, and goes back to Russell Doolittle. Originally it was called the progressive alignment method, where you compare pairs, and in fact that is exactly the way we build up multiple alignments. We take one at a time, compare it to the profile in some way or other. No, I mean something really that is going a step further without really getting another level of complexity. The slides here come from Dariusz Czapulski, who is now at the Broad Institute in Boston, and because it's ultimately originally was his idea, that then later was also taken over from the Zerding group and actually improved upon by the Zerding group. What I show here is CPU and quality. So profile is expensive, so CPU is also essentially a complexity of decision making in some sense. Profile sequence is low CPU, but lower quality, higher profile. What could we do? The answer was already there. You saw it? Okay. So rather than in fact doing a full profile against a profile, what we can do is translate the profile on one end to a consensus sequence. So consensus sequence, now I have different ways of doing that. I could say that in this profile here, I take the one that has the highest odds value. Or I take the majority. Or I take the most surprising value. There are different ways in which I can play that. But ultimately, I will in fact write a sequence that is no longer the one that I observe. So in that sense, it's no longer a sequence of profile, because I'm actually going to change the sequence according to what I observe in the profile. And I'm going to replace the sequence, the actual event sequence, by something that is virtual. The consensus sequence doesn't exist. But the consensus sequence carries implicit information about that profile. And you immediately see So here's one particular solution in which we translate that sequence to that sequence by going here the path of highest. And then we do consensus against profile. The great thing about that is I don't need to change anything. Consensus sequence is just a regular sequence that behaves absolutely in the same way. And in fact, Dariush developed that originally here for PsyBlast. So you run your n number of PsyBlast iterations, you keep the profile, and you just do one more and run the last one with the consensus against the same thing. You don't do a single line of change, you don't do any new math, exactly the same thing, and you put the consensus in. So there is now, let's not get into these. The conclusion is, well, the native PsyBlast, the native blast, so this is pairwise blast, how much PsyBlast improves over that, in some way how the consensus improves over that. So it's almost in some sense the same as you improve over the pairwise. The simple hack allows you a big step, is the conclusion here. And let's just keep it there. Now, the challenge, of course, for this procedure is to, what the slide before said, in fact, it's not that simple. You do have to change the statistics. Because now you, in fact, create a new sequence, and that new sequence has different statistical properties. Because you're more likely to have residues that are more often observed. And you somehow tweak the statistics, and you have to change that underneath in the way you compute the e-value. That was a challenge, but that is doable. Dariush did that. Here's a modern way of doing that, and it's modern because somehow what Dariush did is, I believe, was a great start, but the Söding group generalized it, and here's one of the publications that goes back to a tool that comes from Tübingen, was co-developed with Andrei Lupas in Tübingen. In principle, it's the same thing, except for it is also put into the pipeline of hidden Markov models. Query sequence, query HMM, you have a fast pre-filter, you have an accurate HMM search, which in fact in this particular case here is not really a profile search, but it is a consensus type of sequence. The way they differ from what I just had said before and is that instead of having a 20-letter consensus sequence, which is the same sequence as before, they create a new alphabet of 219 letters. So essentially these letters consider other aspects of a sequence, namely secondary structure and the like, in principle. So you translate your protein sequence into an object that has some features of a protein sequence, and that you put in, and then you do a hidden Markov on this object that has 219 letters. Okay? Procedure is clear? You immediately see now there's a lot of new challenges coming on. Because now, since you have a new dimensionality of the object, you have to completely change the statistics. You have to do this on both ends of it. You have to do it on this profile end, on the consensus sequence to profile end. So there's a lot more work to be done. And then the other part that is a lot of this pipeline here is the way you splice up the universe. So now you don't want to do an all against all, because there are some sequences. So 90% of Uniprot has 55 million sequences. Uniprot had 85 million sequences last year. This year they have 55. Anybody knows why we suddenly for the first time go the other way around? I'm always talking about databases explode. Suddenly we lose 30 million. Why? Any idea? All they did is they threw everything away, or they condensed everything into the same record that has exactly the same sequence. And that reduced by 30 million records. So there is some level of redundancy, mildly put. If you go down, I can't remember the numbers, but if you go down to throw everything away that has more than 90% sequence identity to any pair that has more than 90% sequence identity, I believe you're less than half. So for those, you clearly don't want a profile alignment, because any trivial blast, pairwise blast, immediately handles them, right? So you have to put your 55 million sequences of Uniprot into a hierarchy of classifications, where one hierarchy you look with a simple blast for the simple things. And one hierarchy down there are things where with a PsyBlast you cannot do it. And there you need this a hidden HH blitz. They need the hidden Markov or profile alignment, right? And that is part of this pipeline, and that makes it tricky, and that is a lot of the work that they spend and a lot of the improvement that they do. But ultimately what they show here is that the HH blitz is in fact at every level of iterations much better than Hammer or PsyBlast. PsyBlast is down here. Hammer is the tool that is used for PFAM, and HH blitz is up here. Now there is a new version of Hammer called Jackhammer that is most likely higher. But so some people now, maybe I should include that into our talk. So some groups now begin to use Jackhammer, which is a sort of advanced over Hammer. And in fact, it tries to do a hidden Markov alignment method. It does do a hidden Markov in a way that is different, but in spirit similar to this. And maybe I should introduce that. Okay. Here are examples that you can, with the HH blitz, reach much further than we can do with all of these other methods. So let's review the things we have. Everybody uses PsyBlast. All biologists who publish stuff use essentially cluster W. All these type of methods here, Maxhom, SS search and the like, what the unique view of those is on sequence specific, while these are sort of family specific kind of methods. The same is true for the hidden Markov models, SAM and Hammer. Hammer is in PFAM, and most people use PFAM, so essentially most people use this and that on an everyday base. T-Coffee is more for experts, but it's a remarkable tool once you have some additional information and have the time to really optimize your alignment for your particular case, and you have some additional input, then T-Coffee is the choice. HH blitz, I believe, at this point should possibly replace PsyBlast for the start. There are some issues with HH blitz. In our group, we have had projects where we replaced PsyBlast with HH blitz and some where we don't. Now, again, review where this whole thing is in terms of the sequence space. Sequence, profile gets down here. The twilight zone to the midnight zone. And that's what I'm going to look in a little bit more detail here in the midnight zone. And the question now is, how densely populated is that midnight zone? So I essentially have to do a comparison of all the proteins of known structures against each other. Not quite that. I have to throw away the bias, and then I do a non-biased subset against everything. Okay? So what I'm going to do now, I'm going to do a 3D superposition of all the proteins in the PDB. I'll take a sequence unique subset here, and I'm going to 3D structure superpose everything in the PDB to that sequence unique subset. And then I'm going to ask for everything that is clearly similar in structure, what's the level of sequence identity? Okay? And I'm going to see a distribution. How is that going to look? It could look like this. So there is some realm here where the pairs we have some number of pairs that are very similar, maybe more than this, maybe more like this, and I may have somewhere a maximum here. And this is, just to remind you, the background, random. So if I just take random two strings of protein sequence, they have essentially 20 amino acid by 5, 100 by 5 is 5%. This is essentially the distribution here, right? Okay. Do you believe this is realistic? Okay. There is no assumption. No, it's not. It cannot be realistic because we expect a lot of bias up here. So this is closer to the solution, right? Anybody has any issue with that? Surprised by any part of it? Well, it looks a little riggedy because it's an old version, and again the random is here. Right? I want you to memorize every value in this. This will be asked in the examen. No, this is fake. This is a total invention. This is absolutely wrong. This is not the truth. This is the truth. And the truth comes with a slight additional complication. The truth is always more complicated. So the slice here means that the way I do statistics here is very different from the way it's done here. And the result of that is that this should be actually much higher than this. So this is much higher than that, but if I did the statistics the same way, and there are reasons why I don't do that, then this would be somewhere under the roof. So, and again, you see that skewed here. 25 to 100 is fitting into the same interval as 0 to 25. Is that surprising? What this one says is that the vast majority of all the proteins that have similar structure have levels of sequence similarity, identity, but are in the ballpark of 10%. Mind you, just let's blow up this part here. Red is random. If there wouldn't be any structure, and if there would just be structureless, and that is a random superposition, then that's what I could get, red. What I get from the real structures is this. The dashed one is sort of a fit, a Gaussian fit here. But what I really get is this. The differences between the peaks, in some sense, if you want, is the amount of residues that are specific to realizing all the diversity of structure. And that is in the ballpark of 3% of the residues. That is remarkably small. And not only is it remarkably small, it also suggests that, in fact, if you look at this difference, that, coming from that side, diverging from the identical sequence to getting to the similar structure is as likely as coming from a completely different sequence, at least. So usually convergent and divergent evolution is meant in terms of function. Here I misuse it now, converging to a similar fold, diverging from a same sequence to maintaining the fold. And you see that from the data set we see today, this is so low that it could come from here and there, almost at the same probability. Okay? So we cannot tell. What we can tell is that most likely this implies that, in fact, we had enough time in evolution. Because these 3 to 4%, let's call them anchor residues, that I have absolutely maintained, some fraction of that will be functional. Will in fact not even be there for structure, but will be important for maintaining function. So there is not much more that I can change. And that supposedly means that we reached an equilibrium, that evolution tried all the possible variation it could. And convergent evolution to the similar structure may have been really an important event. And it actually implies that getting really into the midnight zone is almost impossible. This is where, when the similarity that you look for is identical to the similarity of random, end of story, right? Here you can still beat random by doing good profiles. Here you can do much more, but there's a limit to that. We can somehow do it in particular examples. We can get deeper by looking at some regularities and recognize those regularities and learn things from it. And we can use that to, in fact, then possibly, in some sense, this is also what HH Blitz does. Here's just a more explicit way of doing it. You could imagine that similarities between two proteins are not only visible on the level of the sequence, but also on the level of the string of secondary structure and on the string of solvent accessibility. So there's a particular pattern at which residues come in and out, hydrophobicity patterns. And if so, then you could imagine that you could also use a way in which you sort of look at, and that is the idea of threading in principle, where we take 3D structures, we somehow take sequences and align them into these structures and compile some fitness of the structure for sequence, phosphorus. So we simply say, okay, if it adopts that 3-dimensional structure, then that would mean that two positively charged residues will be next to each other. This is unlikely. So this is going to get my energy function down. So David Jones coined the term of threading, or David Jones and Janet Thornton. That means you somehow thread your sequence through the structure, one by one. And okay, I have a clash here, positive, positive. If I moved one, is it still a clash? Yes, it still is. So I move one more. And every single time you compile a structure. In this way, you can use structure to improve your alignment for a protein for which you know the structure, right? This is the idea behind threading, and here the idea behind threading is to simply use a similar concept in principle, but now use secondary structure prediction that we will talk about in 2 weeks from now, and align strings of secondary structure solvent accessibility and amino acid sequence. 3 secondary structure states, 2 solvent accessibility states, 20 amino acids is 120 states. And you do somehow similar as the Söding HH Blitz method. You do an all against all in a different universe of strings, but it's simply dynamic programming. And very early on, this was shown that this works very well. In fact, for many years, this was the best way of doing things. Okay. Now, in order to do this, so I want to benefit from the fact that I can have secondary structure predictions, solvent accessibility predictions, and sequence. And I want to benefit by finding extended similarity, or similarities that from sequence alone I cannot find. What it requires is that, of course, we have to first establish that secondary structure and solvent accessibility is conserved between distant proteins. And I showed you that the midnight zone, so most proteins that have similar 3D structures, in fact, are somewhere down here. But what I didn't show you is boiled down to an identity of states in secondary structure and solvent accessibility. How would that translate to? And you see that here, down to level of 10%, you begin to really lose the similarity. We have now a string of 3 secondary structure states, 2 solvent accessibility, 6 states. And I'm computing the similarity or the identity between the 2 strings, between 2 proteins that I know in terms of 3D structure are the same. But you see there's a lot of diversity. 50% of this 6-state vector differs. Essentially, they look the same. You look at them, they have less than 2.5 angstrom. So you would recognize the similarity. But when you color the secondary structure, you do see that there is a lot of variation, or that the secondary structure rotates, suddenly becomes exposed. So in that 6-state system, you do lose it. In fact, at the level of 30%, you're still pretty much conserved, and then you sort of have a rapid drop here. And that is, again, the red line here shows you 2 structures translated to secondary structure, 2 structures that have been observed. DSSP translation, right? This one here is, on the one, I use the prediction, and on the other, I use the observation. So this is the idea of threading. I come with a sequence for which I do not know the structure. I have in my database a protein of known structure. I want to ask, does that sequence fit in there? Okay? So I compare a predicted secondary structure and solvent accessibility to an observed structure. Right? And the fit is a little bit lower. Is that expected? Sure, we make mistakes. I make, in the black one here, I have prediction mistakes, and prediction mistakes clearly correct in that direction. That much is very clear. So what if I compare to another curve here, which is predicted against predicted? So this is the real thing. This is the predicted versus real. So I have some mistakes and I go down. Predicted will go further down. Sure. Absolutely sure. The funny thing with that is several people, including I, published data in the 90s that the blue line is down here. And then Dariush was just one of those people who was not content just believing his supervisor, but just kept checking and checking and checking for many years and found this is not true. It is... I should not. We never went back and asked why did I make the mistake 5 years before Dariush. I clearly was not the only one, so we had completely overlooked that the blue one was up here. How can that be? Clearly, now I'm comparing 2 predictions, and clearly both have mistakes. How can they be more similar to each other? They're correlated, and somehow we'll put it slightly different. Let me just say the opposite. You're on the right track. It's just a little bit the other way around. The ones where they are wrong is with the regions where it's not that important. So they make different mistakes in some sense, but where they are correlated, that's where they are more right. And that's where the structure really is importantly conserved without the dynamics. Where they make mistakes is more movement. That's where the red ones also differ. Where they are consistent is where movement is less. And that brings this number higher, which is a remarkable finding. Darsh, thanks for the slides, and that you can use to improve the prediction PsyBlast, and then use something like this here. You can do much better. Okay, so let's go to comparative modeling. Again, on next Thursday, oh, that is the wrong, that's the wrong date, that's the wrong slide. Next Thursday, there is no lecture. There's a Corpus Christi. Thanks for Andrea for some of the slides that are to follow. Comparative modeling, again, so let's just go quickly through this. Sequence determines structure. In principle, we should be able to predict structure from sequence alone. Here we have KRAS, an oncogen, the structure where the coloring here is simply according to N to C term. Now we have a mutation. The way this reads is glycine at position 12 is mutated into cysteine. And these two look very similar, even in the binding site. Let's look at the rash, which is lime, and the green is now the RAS. So these two have 85% sequence identity. And you still, you essentially, you see a little bit that the helix moves here. This helix is a little bit wound differently. This helix is absolutely identical. The strands are aligning very well. Binding site is similar. We go one down. We go to the level of fly. So the first two were still fly. And what I compare here is all proteins of known, all experimental structures. Fly, 28%. Again, the slides come from Andrea. In orange, and now you begin to see, we still actually have this sort of helix, but you begin to see that the binding site is changing. You begin to see that some things are really changing, but this is at 28%. So we are already evolutionary, we are between fly and human. We already have some distance here, and we essentially still have exactly the same structures, right? Let's go into bacteria. Now we actually lose so far RAP, RAS, RASH. We have some similarity in the enzymatic function. Here in the bacterium we lose that one too. We now have 19% to human in the purple here, and you still essentially recognize the fold. So essentially still the same. And this ultimately is the background behind comparative modeling, that you in fact try to identify the similarities from sequence alone. Let's put another perspective into this. Can we use structure to predict function? So here's one example, conserve. Conserve from the Bental's group in Tel Aviv. All you do is you take a structure and you label on the structure what you look up in the sequence alignment as conserved. Okay? And this conservation, where dark red is conserved, light blue or dark blue is variable, you see that the dark here is exactly where the binding sites are. So just taking the structure, looking at the label of conservation immediately guides you to understanding something about function. And that's remarkable. It's extremely simple. And it's back to the question, why do we care for structure? Because of conserve is essentially one example for that. Here's another thing that is important. So here we have two different proteins with a different electrostatic surface. The structure underneath this is the same. In this particular case, and that's Diane Murray who gave me that slide, in this particular case here, one of them, and I cannot remember which one, yes I do remember, this one here, actually is a comparative model of that one. So you use essentially the same structure, but put a different sequence in it, and you get a different electrostatic surface, and immediately guess that the function will be different. Again, this is something that you cannot do if you would not have a structure prediction. And then there's another aspect on a larger level to really get complexes together. But let's get back to the modeling task. The modeling task is to simply find for your sequence for which you don't know the structure, which of these shapes that it's going to adopt. How can we do this today? Let me give some numbers, and these numbers are outdated. But I haven't done them more recently. And the idea essentially is the same. Here is the numbers translated to today, but the actual computation underneath was done in 99. So Uniprot was then 35 million, Swissprot 400,000, PDB about 100,000. Roughly, this is 55, this is half under 10, and this is still 400,000. Now, let's get to the redundancy in PDB. The 100,000, most of it is here. I mean, a lot of the reduction of, this is when PDB was 70,000. You see that just throwing away everything that is 100%, like the story for Uniprot that I told you about, is almost halving. Why is that the case? Why are there so many things in the PDB that essentially is the same structure again? That's one reason. Absolutely true, and it's an important one. It's an important driver. In some sense, we may call it a negative reason, except for, on average, the one who comes second is better. The structure is better. So we improve over the structures, and that is a good reason. Another very good reason is that, in fact, they study the same protein with different ligands in different situations, with or without the other protein bound, as a monomer or as a dimer, with or without calcium. And that, again, is a lot of what is functionally important to study about the structure, and explains this jump here. But anyway, so we have a certain reduction. Now when we do homology modeling, again, the idea is we start with a sequence, we find a similar protein in the PDB. Once I find that, I assume these two have similar structure. And again, the types of tool I use, the types of sequence comparison, sequence, profile. I can do that. So experimentally structure known in some sense, you can translate this here on the Uniprot base to 13%. So this is, well, it's an overstatement. For 22, you can sort of do homology modeling. Okay? Homology modeling is by simply going above this curve. Everything above the curve, you can do homology modeling, and then that's sort of what you get. Then you go a little bit further down into the twilight zone. I showed you that in the twilight zone you have an explosion of the red, the negative. You have an increase of the blue, that's why you want to enter, but it's more complicated. You use profile. You get a little bit further down, you add this one to the pi. Then we get into the midnight zone, so we cannot reach there, we cannot get there, and that in some sense is the end of the pi. For everything else, all we can do here is 2D predictions or 1D predictions, and that is what I will talk about in 2 weeks from now. For today, let's relook at this pi here. This pi is a pi done on the databases that we have. Databases are biased. How could you imagine that we get a version of the database that is less biased? Which is also a database, but which, if I told you, okay, if you gave me Swissprot 400,000, I don't want that because it's too biased, or 55 million Uniprot is too biased. How would you try to give me something to make your supervisor happy? What would you throw at me? Can you think about a subset that is less biased? Well, one way in which we could do it is just an unbiased subset. But that, in some sense, is not natural, in some sense, because it doesn't occur in nature. What would be an example for a set of proteins that occur in nature, that brings some bias and avoids some other bias? The genome, right? I can take an entirely sequenced organism. Human. All the proteins in human, there's some bias in there because we have duplication, we have many duplications event. Certain proteins are more duplicated than others, so there's a lot of bias. But it's not that people study mouse, fly, and again, do the same structure again in different organisms, and do the experimental work in different organisms simply because model organisms are easier to do. So that bias at least is gone. And if I look at that, the genome view looks a little bit more like this. So comparative modeling is no longer really covering much more than, well, it's certainly not reaching more than half. The good news is, just sit there and wait. Things get better. So this is the view from 91 to 99. And again, this the non-genome view is the switchplot view. This increase here has nothing to do with improving methods, just because databases grow. That's all. And the database of sequences and structure. Okay, let's just look at another view here that comes from Jinfeng Liu. And this is essentially the view I showed you before, and I'm going to zip over that. Let's get back to concepts. So beginning with lingo, the language. Comparative modeling versus homology modeling. Historically, people used homology modeling because they argued there is an evolutionary relation behind. So I can infer the similarity in structure because there is a connection. Ultimately, that is not quite what we do today, and it's not quite right. We compare because what we can compare has similar sequences, and similar sequences follow similar 3D structures. Ultimately, that's the point that gave this word comparative modeling. There's a target and a template. Target is the protein to model. Template is the protein to model from. So template is the protein in the PDB, and that's my query. The target, right? And again, so we have three different, that's target, two different templates, and I simply find the most similar one. The first step, therefore, is identify the template. How do I do that? Well, standard blast, depending on the similarity, I simply find the closest hit, pairwise blast, or more. Then I have to align. Align makes some mistakes. These mistakes cannot be really corrected. Then I build a model. How do I build a model? So I now have my query sequence, my target. I have a template, I have a structure, I have a protein of known structure. How can I build a model? Give me one solution. The simplest. Whatever you find in your head is the simplest for you anyway. Well, you just simplest solution is you copy the coordinates, right? The alignment implies a corresponding position, and for the corresponding position, you have a C here, an A here. You just look up that A and read the 3D structure. Read the coordinates of that A and put it onto your C. Right? That's the initial model. That initial model makes some mistakes, maybe. So you assess the model. How can you assess the model? Well, it's missing here. So you could, in this particular case, see whether this would imply that you would pull the helix too much apart. Then it's not a good idea, and you try to move a little bit back. And things like that. Then you try to refine it. That's exactly what I mean. You can try to stretch or move back to adjust it to that situation. What are the challenges? Well, up here, when two sequences are very, very similar, it's the speed of modeling. At some point it becomes the quality of the model, then the problem becomes the alignment, and further down here really becomes to find the protein that you in fact model. Aligning now these days is done by HH Blitz or profile methods, and this is sort of the variety in terms of accuracy here. It's the variety of levels of accuracy that you can reach. This is a nice overview paper from David Baker and Andrei Shali. Let's get into state-of-the-art methods. Anybody can name some? Have you ever heard of comparative modeling methods? Okay, here's number one. Modeler from the Shali lab, your CSF. This is Andrei Shali. Did his PhD in Birkbeck with Tom Blundell. Went to Harvard Medical, Martin Karplus, Nobel Prize. Went on to become professor at Rockefeller University and then at UCSF. Over 400 publications, 1 over 4,000 times, H-index 64, clearly a giant in the field. The modeler publication, the first one here was with Tom Blundell in 22 years ago, and that is a more recent version in which Marc-Martin Renom put the whole thing into a big package. Modeler is an immensely big package. Here's the website that you can use, you run on your computer, and it has essentially all the bells and whistles if you really want to build a nice model. Modeler needs a registration, it needs a download. It's not the thing that you do for 10 minutes modeling or 5 minutes modeling. These days, Modeler actually has a website where you can also look up pre-computed similarities. But still, Modeler, the absolute strength of Modeler is in you using the interface and everything that comes with it. It is for free, and it's an interesting way of doing it. Let's just look at some of the elements of what they do. So again, the first problem, of course, is find which PDB structure you want to take, then align, then build the model, evaluate the model. Whenever you say model is okay, you stop. If not, you get into different iterations here, where this one could be maybe change the alignment. Here, maybe try another template from the beginning. So let's get it through some of these steps. The constraint satisfaction here, we have constraints in 3D, and you have a particular model that you build, and that model has to satisfy those constraints. So you see certain C alpha distances. I said copy the coordinates. What if these coordinates suddenly mean that you have to pull the C alpha apart? That cannot be true, right? So how would you make the model, how would you change the model? Well, you cannot really move the C alpha back to the position so that it fits. You have to do that in some sort of probability value. And you have to sort of begin for the first pair where they clash, you move them a little bit closer. And you immediately see that will move the rest a little bit apart. So you have to iterate over that and find a molecular dynamic simulation that in fact optimizes this path through it such that you fit to the observed density. Essentially that's what this says, and you do a certain number of iterations, and the further you go down, the more likely you will find a stable solution that in fact fits all the constraints. Then there are the objective functions. What that again means in the alignment field is that the two strings have identical residues or something like that, right? A percentage sequence identity is one objective function. Here the objective function is a little bit more complicated. So when we discussed about how to compare two proteins, we had the objective function of RMSD. And there are other objective functions you can imagine for 3D structures. The problem here is that we have a universe where sequence actually has changed. And we are not comparing three-dimensional objects. We are comparing fit of a sequence for a structure. In some sense, it's the same problem as that I mentioned in the threading. And there are different ways. You could have a molecular dynamics, you can have Longevin dynamics, and other ways. You can have rigid body superpositions, rigid molecular dynamics, rigid minimalization. Modeler is around a long time. So essentially, they tried all of these options, found advantages in all of them, and somehow what you get is a package where you can choose, and by default it finds a path through the choice of options they have. There are fluctuations, and these fluctations help you to in fact explore local minima. Local minima mean you know in which direction you move. Okay, C alpha, C beta fit to the model. Sidechain packing is a typical error, so that the structure is leading to a clash of the sidechain. That, in principle, is still relatively easy to correct. With sidechains, you can move around. What is more complicated here if you have a misalignment. If you simply say that the cysteine corresponds to that alanine, but it's wrong. So that means that you in the helix would have the wrong register, or that you simply have the very wrong point. The corresponding point might be a different one, and that you cannot recover. Or how could you recover that?
Correct alignment.
Yes, and that is, but you immediately see what it will do. You will open a can. So you will then begin to assess, essentially you have a new alignment function. If you want, this is like writing a genetic algorithm with a model in mind. If you had the CPU to do that, that would most likely be the right way of doing it. Trouble is, it takes a long time. Because the moment you find a new alignment, you have to again energy minimize the constraint satisfaction, or thousands of iterations. So this is really bulky. But yeah, that is what they, in some sense, they try. Where there are very obvious alternatives, they try them. If it's entirely the wrong template, you hope that you will see that because the energy function will blow up. That something in your computation will tell you this model really is wrong. But there's no way you can recover from that other than you have another template and say, okay, I choose that one over that one. And again, Modeler does that. It gives you, it essentially uses multiple templates in this, I believe one of my next slides, in two different ways, where one way is to somehow find a path that is in between the two, and one other way, the more standard way, is to find the one that is better fitting for me, okay? And so you hope that one of them would then be wrong and you would see that the other one is right. There's a score that it has that essentially is the optimization of the modeling procedure. It's related to pairwise energy. The low dope score here means a lower RMSD. So mostly you see that there are exceptions to that, but mostly you see, so the lowest RMSD value is this one here. This is not the lowest score. What you would pick is this model, but this model still is a fairly good RMSD, right? So the internal energy score, and here are three different views of that. The internal energy score is a good proxy for getting the right model, but is by no means always right. So if you know the answer, you could always do better. Trouble is you don't. And in most cases, you do find a quite okay model. But that again, so getting it to this point here, this is 20 years of work for a group that has been busy with several people. What if the loop were missing here? Can we model a loop? So in this particular case here, we have two similar loops between two proteins, one in fly and one in human. So we have a slightly different loop. But what if the loop really is completely missing? Can we do that? Well, in principle, this is like the problem of protein structure prediction. Now, it's simplified in the way that rather than having to model an entire protein, I only have a loop of 10 residues or 5 residues, and the loop is constrained. I know that this is clear, this is clear. All I ask is how the thing in the middle goes, right? And we could do that by molecular dynamics. We could hope to do that. Actually, and that's a publication from Anders Fischer, where the loop length is shown versus the RMSD. You see 2 angstrom is a sort of okay number here. For loops until 6, 7, this works, but after that it blows up. 6 angstrom for a loop of 8 residue is essentially meaning that you have a very different orientation. We cannot do that. So for short loops, yes. For long loops, doesn't work. Okay. Modeler. The other way is Swiss model here. Today from the Schwede lab, that's Thorsten Schwede in Basel. He actually, Thorsten did his PhD in X-ray crystallography. He is an experimentalist. Few publications, but very visible ones in there, and amazing web presence. So the work they do in their lab has over 1,000 accesses every day. It goes back to an idea from Manuel Peitsch, who's now at Philip Morris, and Nicolas Gouet. I believe he's now in Lausanne University, I have to look this up, published in some obscure journal. And that idea was, I'll get back to that in a minute, it was then refined by what Thorsten is keeping up today in the Biocentrum in Basel. The initial idea, here's the website, the initial idea, let's call it the underlying philosophy of Swiss model, was to have something that is fully automated, that is for non-experts. And then Manuel had this idea, the less you do, the fewer mistakes you make. So what that actually entitles is, you just copy the coordinates, job done. There will be positions where you have clashes, leave it to the user to find out they exist. leave it to the user to do the right thing with it. They are experts, they are biologists. I'm a computational biologist, what do I know? That in some sense, Manuel is a very clever person. If I don't care, then I don't worry, and then I can put out something that really is simple. So the original method really was an alignment by blast and psi-blast, copy the coordinates, publish on the web. That, in fact, was the first method that had an online version of comparative modeling for everybody to use. The shock for the field was that it performed extremely well in CASP. So well that people began to use the server instead of their own methods. So this stupid, simple thing was actually on par with what people had spent years on developing. That was very frustrating. But things got more complicated from there. So today, Swiss model is way more. It's much more like Modeler is a package that you get with all kinds of additional tools. Christopher Andrew Wilton, some of the slides I take are from him. It begins by choosing the type of search that you have. So they have HH Blitz in there now. You can use different alignments. You can use different parameters. There is a version where you can have pre-computed comparative models for everything. But then when you come with your own sequence, you suddenly face a web page where you have to choose a lot of parameters. You see secondary structure, you see sequences, you see families. You can use all of that to steer around the way you go on. You have a loop modeling module, you have a core module, you have sidechain placement models, you have energy minimization. In this particular case from Homos, steepest descent is similar to what Charlie does, what Modeler does, but in detail slightly different. And you have a visual interface that looks like this, where you also here see the 5 psi angles. 5 psi angles essentially tell you whether you are in the right space, so you want a population here, some population here. If you suddenly observe the population here, you would see that something is wrong. But you immediately see this is already a tool for more experts. So in order to see the value of that, you need to be a little bit more of an expert user. And this is no longer really the initial simple tool that it once was. There is tons of packaging, molecular modeling packages underneath. There are, at the end of the day, you have a residue, you have some energy scoring by residue, some average, and this is essentially what you get as an answer for how good is your model. Okay? How good is it? So different colors here. Green is sheets. The pink here is a helix. The black one is the none of the above. That's per segment averaging. Per residue averaging, you see a lot of variation. So what you essentially see is there are regions where it looks pretty okay. And there are regions where, in fact, it doesn't look so well. It is better where it's in the core. The functional residues, however, are above zero. So functional residues are less well modeled in this particular case. And you have to really now get into the point where you ask yourself how well this model maps. You have to ask, what does it imply in terms of function? In my model, where do I, from my model, what would I say where the binding site is? What would my model say about how the binding mode will operate? And that's when you begin to sort of investigate. That's when you begin to look at these detailed plots where you ask, that is my binding site. Is that a site where I have a lot of biasing, a lot of unclarity about my model? Okay, Swiss model was once completely automated, still is a version automated, but today is much more flexible, and these two packages grow more and more together. Other comparative modeling methods, H.H. Pratt, in fact, I'm going to quickly go through all of these. H.H. Pratt coming from Johannes Söding and André Lupas, which in fact, in principle, the major step here is to do the alignment differently. Essentially uses HH Blitz, that one CASP, so then Swiss model copied it, And this is the way the field evolves. So somebody does something interesting that works, everybody else copies it, and things get more and more similar to each other. At the end of the day, still important differences. The major steps you build is really alignment based, and that essentially most of this is what I already discussed before. It still picks the best template, and at this point here you see immediately the similarity of a method that HHPrep1 casts at various instances, and at the end of the day, at the most important point, it says run modeler. So we're back again to the same story. So really the big differences comes in the alignment here. A 3D jigsaw from Paul Bates and Michael Sternberg. That idea really is you get beyond the idea that you have a core of a structure that you want to match. Rather, you have bits and pieces, like a jigsaw puzzle. So you have local motifs. It could be a helix-turn-helix motif. It could be a sheet, so non-local in sequence, that you simply cut out. And you want to see whether what you build as a model fits into all the space of structures. And one way in which you could do that is you simply take the PDB, put it into jigsaws, you cut out your little pieces, and then you take your protein of unknown structure and see whether it fits to which piece it fits, and then put the pieces together. Another way of calling that, so they were really the first to coin the terminology. At the same time, others used the fragment assembly terminology. It's a similar idea. So it's a fragment-based approach. We identify the similar fragments in the PDB, assemble the fragments to the structure. And obviously, all the detail is in how this is done. But that's the idea. What if, the background to that, that's Gert Friend from Nijmegen, and Yasara is from Elmar Krieger, is simply the visual interface to what if. I'm not saying much about what if. What if started with a simple question. If I now have a model, and something is a clash in the model, or if I want to, in fact, change this to a different residue, what would happen? How would the model be influenced? Something changed. So in the context of doing engineering, so you want to engineer an enzyme to do something better, to cut a particular bond easier, or be more resistant against heat, or whatever your goal. What in my model do I have to change? What in my structure do I have to change? What if was the idea I changed that particular residue? What if I changed that bond? And the goal was simply to develop a package that would answer that question. The result of that is, I don't know, at some point it had 1 million lines of source code. It's something that Gert has been doing. This is I'm not entirely sure, I quoted, I believe it was the first publication 25 years ago. But at that point he had already been working on it. So at that point I would venture to argue it had already been more than 100,000 lines 25 years ago. And essentially he has spent every day in his life and written another 500 lines of code, I guess. And it grew and does play chess, and it does really play chess. It also plays Go. It has all kinds of Easter eggs in it. And you know, like always when a particular person develops such a big package, it's also very much shaped on Gert's head. I mean, it doesn't, the package doesn't look like him, but it thinks like him. So the way you interface that program, you have to think the way Gert thinks. And that is where Elmar Krieger came in and said, well, let's just put it in a user interface, something on top of what if that is more simple for people to use, and for instance, produces graphics that are nice. And a lot of values in graphics. These pictures look nice, very easy to do. I mean, it's not that easy, but it's easier than rewriting what if or completely changing some small subroutine in what if. Easier, it's not easier, it's just maybe the same complexity, but this is immediately visible. Looks like a more fun project. Okay, so there are different types of users, different types of solutions. Ultimately, they cater to your perspective. Coma is, again, that comes now from Czesław Wędrowicz, and his special contribution is in the way he does the alignment, and mostly he does it through expert filtered profile alignments that are done good. And he's very good at distinguishing right from wrong. He's very good at, in fact, looking at models and discarding things that are wrong. That led somehow to Coma, and Coma is another method out there that competes. ITaser, now we get into the realm of more the CASP winners, Zhang. In fact, that is an automated method that does it all and predicts from sequence on structures. Most of what ITaser does is really getting down to comparative modeling. So structure prediction today is most accurate for comparative modeling. The workflow, like everybody else, you do a structure assembly, put together the contiguous fragment, so have a knowledge-based force field that simply tells you whether what you put together in those fragments is right or not. You have some spatial constraints. I showed it first today with Modeler. You have knowledge-based statistics terms, that is essentially the bonding sequence contact predictions, so you predict system bridges and the like from sequence. I'll get back to that in 3 weeks or something like that. And use these contact predictions to match it to your assembly. You have multiple domains, you dock different models, you try them at the same time, you have multiple templates, you assemble them all, and you optimize global and local structural packing. And that again is a very useful method. I haven't tried it, I don't know to which extent it's available, but clearly it's one of the top players out there. Okay, let's wrap it up. The most important thing is in the title, you show me the mistake of your method. Here's the background. When in Magritte, ceci n'est pas une pipe, you may understand this is not a pipe. Is it? What does it mean? I understand that this is a class about protein structure prediction. It was not initially meant to be art interpretation.
This is a picture about the pipe.
Exactly. So this is just a photo or a painting of a pipe. It's not the real thing. And that is in fact the point that you have to remember. When models and reality, ultimately model must be wrong in some respects. Otherwise it would be not a model, it would be the thing itself. Then, just like this, it would be the pipe, not the picture of the pipe. A model has mistakes. And this, in fact, is what Thorsten says. A model is a tool that helps to interpret biochemical data. Model does not have any right or wrong. There's no, so far what I talked about is in CASP, what we check is how well these comparative models do in order to predict structure. Ultimately, that is not the question. What we want to do is we want to predict function. What we want to do is learn from the model about our sequence to do another experiment. And for that, we want to look at something else. And that has to do, maybe when the model is totally wrong in most places, I can live with it as long as the binding site is right. Or as long as it tells me something about how to do the next experiment. One contact that I guess from the model might be enough to do the next mutation that knocks out the interaction. Right? The value of a model is somewhere else. And the value of the model essentially is in the eye of the beholder, is in what you make of it. And that begins always from the question, where is my model right? Where is it wrong? Whatever prediction method, if you use it, you have to, as a developer, you have to provide tools to the user to make the difference. Where is it right? Where is it wrong? For somebody who uses it, you know this is not the real thing. It's a model, it's a mistake. So when you look at it, ask yourself first, where am I more right? Where am I more wrong? Let's call it an exercise. When you look more general in terms of scientific projects, when you plot data, there's always an outlier. Go there first. Try to see what the outlier means. Maybe it's meaningless. Maybe it's just an outlier because there's a distribution. Maybe it's a mistake in the program. And maybe it's telling you something. Not a mistake, maybe it's a limitation of your approach. And most of these findings will bring you somewhere else. So state that, find that, pursue that. On a model, ask yourself what is right, what is wrong. Why is the model not reality? Next week we will go into secondary structure prediction, I believe. Yes. I talked about the assignment of secondary structure. I'm getting confused here. I talked about the assignment of secondary structure, right? Didn't I? From 3D structure?